today what we're going to be talking about is the foxp3 protein / gene. So the foxp3 protein, which also acts as a gene, is responsible for cellular response, really. So let's say this is the foxp3 gene, and then this is a Th17 cell. So if that connects to the Th17 cell, it influences the immune responses. So it controls it and modulates it. So it can decrease the amount of inflammatory cytokines in the body, the amount of inflammation in the body in general. This is known as the four-head box. In fact, it's actually very important because scurvy mice, they're mice with autoimmune diseases. If they lack the foxp3 genes, and so they develop severe autoimmune diseases which lead to death normally. So the environment is also important in the foxp3 genes, because the foxp3 T cells, should I say, because depending on what environment it is in, it can change into either a T regulatory cell or a Th17 cell. So what I'm going to say is the presence of interleukin 6 and TGF-beta, beta 1, changes the naive CD4 cell. This is a CD4 cell, I believe. It can change that into a Th17 cell, which I've said is very inflammatory, because the Th17 cell has is linked to the overproduction of interleukin 17, which has models that are based in like the autoimmune conditions like MS, rheumatoid arthritis, and IBD, which is also known as IBS. So the thing, a thought about it is that why don't I just take someone's foxp3 T regulatory cells and put it into my body, for example. The problem with this is that 50% of the cells get converted into foxp3 negative T cells, and then after that, that gets converted into interleukin 2, interferon gamma, and interleukin 17 producing cells. So we don't want to produce interleukin 17. So it's, even though interleukin 2 is actually advantageous, because interleukin 2 leads to an increase in foxp3 T cells, which lead to T regulatory cells being created. But the interleukin 17 is more, let's just say, it's stronger. So it's like it's too strong compared to interleukin 2. So the benefits of the interleukin 2 is basically nullified by it. Depending on the environment, the CD4 naive T cells convert into T regulatory cells or Th17 cells. So environment high in, I've already said this. I've basically already said that. Vitamin A, or atra, which is a variant of retinoic acid, can decrease the numbers of T effector or T killer cells by reducing their interleukins of interferon gamma and interleukin 4. So what I'm going to do now let's talk about a study to like prove my point, if that makes sense. So they found that ATRA, when ATRA plus TGF beta 1 was administered, it caused an increase to about 35% proportion of T regulatory cells, or which is also known as foxp3 positive cells. But when interleukin was added, so let's just say plus IL-2, then there was a 90% proportion of that was 90% proportion of CD, no, it's not CD, it's foxp3 positive cells, yeah, foxp3 positive cells, which just shows how anti-inflammatory it is. So if you have a 90% proportion, like it's obvious that the inflammation will definitely decrease, especially when it's, let's say if it's even roughly 100%, which is the sake of it, then there will literally be no inflammation going on in the body, which is hard to do, but it's a good step in the direction of finding remedies to sort out autoimmune conditions. What I'm also going to talk about is the retinoic acid receptor. So for example, TH17 has a retinoic acid receptor that's called ROR gamma T receptor. So when exposed to interleukin 6, it increases the number of these receptors, right? Which kind of shows that it's kind of yearning for more vitamin A or retinoic acid. But when retinoic acid is present, it starts to downregulate these receptors, which can essentially lead to more control of the inflammation as well. So there's multiple mechanisms in how retinoic acid plus interferon 2 can be used to get rid of autoimmune diseases as well. But it just shows that the foxp3 gene is very relevant in trying to deal with autoimmune diseases.